All right, moving on to 26. So, paternity suit, right? We want to know who the dad is. So what they'll do is look at different alleles, different gene fragments that tend to be different between individuals, so that those differences can be picked up and a paternity can be established. So they look at one locus, which is one certain gene, and there's two alleles. And you can see there's this 3 codon allele, and then this allele has 5 codons. Okay, gel electrophoresis was used, and as you know, which one should go further? Of course, you should say immediately the B3 is smaller, lighter, less mass, less friction, can go through the gel much faster. So band 2 is going to be the smaller one. That's going to be 3. Band 3 is going to be band 1's larger. It's not going to go as far. So that's B5, B3. And down here, band 1 is only going to be B5. Band 2 is only going to be B5. B3. Remember, it's not going to be a mix because gel electrophoresis is the whole point. The whole idea of it. Now, you guys have done it twice, I think. At least twice. 3 times, maybe. Some of you. It separates by size. That's the whole idea of it. 27. Got a nice big diagram showing this piece of DNA getting modified, or RNA being modified, sorry. Even I get them mixed up, obviously. So we have parts that are expressed, exons. We have parts that aren't expressed, non-coding, called introns. And those are taken out. So the coding area is maintained. This is kind of confusing. I think intron, it means A, it should stay in, right? It's in. No. And exit should leave. Nope. Exit is reverse, and that's because it stands for expressed. So W, that would be an exon, and X would be an intron. Okay? So RNA processing, which remember, only happens for eukaryotes. Prokaryotes, bacteria, they don't do all this funny stuff with their RNA as they're processing it. Ooh, got a lot of nice diagrams for you here. So, protein structure, all these different primary, secondary, tertiary. What kind of interactions are found in secondary but not tertiary? And that would be hydrogen bonds. That's the only one of that list, because ionic, disulfide, very characteristic of a tertiary structure. Hydrogen bonding does happen between beta pleated sheets, that's my elbow, and alpha helices in that secondary structure. Okay, remember quaternary structure down here. I tried to draw several different polypeptides together combined. Okay, make sure you review this by writing those diagrams down. Mitochondria, the powerhouse. Where would ATP synthase be? So remember, you got 2 layers, right? So 2 layers of membranes, or a double membrane. In the very inside, you have the matrix. This is the intermembrane space. And the synthase, that's what I kind of tried to draw here. I'm going to highlight it. That's it. That is right along that inner membrane, because it's going to be pumping out hydrogen into that inner membrane space. All these electrons are going to be, or electrons are going to be building up in the inner membrane space and then passing back through, changing ADP back into ATP. All right. So ATP synthase is along that that inner membrane. Okay. Inner membrane. Not along the outside, not in that space, not inside. Okay, right on there. So along inner membrane, like I wrote. Which space has the highest hydrogen concentration? That's inside the thylakoid space. And that's because the sunlight comes in and breaks that water up, oxygen leaves, and then you have a whole bunch of hydrogen sitting around inside the thylakoid. So remember, you have two basic areas, spaces inside the chloroplast. Inside the thylakoid and then outside the thylakoid. And that area is called the stroma, outside the thylakoid. So we got inside the thylakoid and then outside the thylakoid in the stroma. So we just did a warm-up on this about competitive and then non-competitive inhibition. We know that competitive inhibition is less restrictive, has less of an impact as non-competitive. Non-competitive has a bigger negative inhibitory impact on the enzyme action because it just shuts it down. The competitive inhibitor and the substrate are competing for that active site, and sometimes the substrate makes it, sometimes the inhibitor makes it. So that's basically what this is asking you about. Why? I think I covered it. That's why right there. Competitive, and this is non-competitive down here. Water uptake by roots. You should know that it's osmosis, and that solute concentration in the root is greater than in the soil. Remember, water moves towards a higher solute concentration, like a saltier environment. Think of it that way. Wherever it's saltier, it's going to move there more easily, more readily. Okay, oh, beautiful. I love flowers. Mother's Day, a couple days away. So I got my wife some flowers, my mother some flowers, but I'm going to enjoy them just as much as they are. So on your paper, please label everything. All the parts of the flower, you should know this, or at least be able to, manage your way through it. And this is just, memorization. Where is that Y? Yeah, the ovule, the ovary. Ovule is the seed itself, and then the filament holding the anther. These are the male parts of the flower with the pollen, and the middle parts are the female parts, as they're termed, where the pollen goes to the stigma, grows a pollen tube, and then fertilizes the ovule inside. Positive phototropism. Plants can bend towards the light. Their light gets shut off. They can grow up to access more light, and that is auxin, and that's making the cell elongate. And if it goes on the shady side, it'll bend towards the light. Big diagram talking about crossover. So this is, I went ahead and annotated this. This is A. Both of these are going to be big A, big A. This is A. Remember, this is a copy of this, right? So these are both big A. These are both little a. Same thing down here. Both big B, both little b. But remember, these switched. So this little b came over here. This big B came over there. And the only one that matches is down here with D. Okay, showing that crossing over event occurring here, being expressed, or seeing the result down here. Sometimes, I've noticed IB, I don't know, it's just how you read it, but sometimes the bigger the the question, the easier it is. But in your mind, it can be the opposite. You think it's a big, huge thing. It's going to be brutal. But just stick with it and do your best. Use the diagram. Like this diagram, you can kind of figure out if you don't remember so well about crossing over and how genes work. This you have to kind of remember. This you could kind of figure out because you might, the shady side, it's going to go this side, it's going to bend it towards. So try to use your reasoning best you can to figure it out at all times. Alright, reproductive isolation after one generation, that would be polyploidy. And the reason is that involves the doubling of chromosomes. And if you double the chromosomes, you're not going to be able to breed. Remember, our organisms have real trouble if you mess with the number of chromosomes. Down syndrome, just an extra chromosome, causes major problems. None of these other things are going to cause reproductive isolation after one generation. Okay, and polyploidy, all these extra chromosomes, that tends to make the fruit get bigger. So a lot like the strawberries that you eat are all polyploid, and that means they have 56 chromosomes. They should have like 8, but they're like tetraploid, so they doubled and doubled and doubled, and that kind of made it bigger and bigger. And we like big strawberries. I wonder what it would be like to eat a little strawberry. I don't know. Maybe it's better. So because of COVID, we know about our immune response responding to antigens. The secondary response is important after you get that first exposure because of your memory B cells. But sometimes some viruses, you don't get a secondary immune response, and that is because of that very high mutation rate, which is why you need a yearly flu shot, which is why we needed COVID boosters as we progress through that pandemic. Who knows, maybe we'll have another one. What hormone is inhibited during pregnancy so you don't have contractions? That would be oxytocin. That is the contraction hormone. Okay? Oxytocin. You have too much oxytocin before the due date, you can have a miscarriage. Alright, here we have a kidney. I think probably some of the more complex content that we have. So, it shows what this blood plasma is like. And again, this is kind of, it's not as difficult as it might seem on the outside, or at least on initial review. So we see the chloride ions, it's the same plasma, the blood plasma, and that might confuse you. That's basically your blood. And the fluid inside the nephron. And that eventually, inside the nephron leads to kidneys. Leads to the bladder. This is already in your kidney. So this is going to be leaving stuff here. If it stays here, it's going to be leaving your body. So the chloride ions are the same. Glucose, the same. Urea, the same. Proteins, still not very much protein. Most of those proteins are in the blood, so very little, but similar blood. And the question is, which section would you expect to find these concentrations? That would be in the Bowman's capsule, because this is where everything is coming out of your blood. Almost everything except the big stuff. So all the concentrations of these small ions like glucose, or ions like chloride, then smaller molecules like glucose and urea, they're going to be about equal here at the Bowman's capsule. Okay. Now in the proximal convoluted tubule, that's where you have things being reabsorbed. So your chloride ion is going to be less, a lot less. Same with glucose. Urea is going to be about the same. Maybe more concentration because you're going to be losing water and other things. Because your body's going to want to get the glucose. You can't pee sugar. It's not going to be good. Or chloride, you're not going to be able to maintain your heart rhythm and other vital processes. And what I want you to do is just kind of diagram this or annotate this, just to remind yourself of what happens at all these points. Most of your stuff is reabsorbed in that proximal convoluted tubule. You do some more absorption later on, especially of water, but most of the essential things are reabsorbed. And collecting duct, this whole question with ADH, antidiuretic hormone, comes up. This is where your body can conserve water when it needs to, get rid of water when it doesn't. So if you drink a whole bunch of water, you're not going to have much ADH. But if you aren't drinking, if you're exercising, your body's going to make more ADH, and that's going to make you suck more water back out from the collecting duct to keep it from going into your bladder and then, losing it. Last question. So, here we have the placenta, maternal blood, and fetal blood, and the direction different substances are moving. And the question is, which one requires endocytosis? And you should remember, this is for large molecules. They're too big to fit through the protein channels that your membranes have. Okay, so if you look at these, water, it's small. Oxygen, even smaller. Glucose, pretty small. Urea, pretty small. Immunoglobulins, those are proteins of your immune system. And those are pretty big. At least compared to the others. So too large for the membrane transport protein. So that'll be D. So that completes everything for 2017 Higher Level Paper 1.